So I'm going to kind of split the lecture. We're going to do a little bit of STEM anatomy so that you are up and running for when you walk into lab today. You're going to be looking at a variety of stems and working with stem tissue. And then I'll switch over to the photosynthesis material. There's a few last things to go over, and I'm going to do another review on that. Okay, so stem tissue. All right, now we're working our way up from the roots, and you see two images here. One is a dicot and one is a monocot type stem. And so first of all, when you think about monocot roots, we saw that there was a ring of vascular tissue in the monocot root. Now here with the stem, you're seeing a ring of vascular tissue again, up top there, ring of vascular tissue, but that's actually a dicot stem. So that's a little confusing, but I'll try to convince you of how this happens. One way that I can tell that this is stem tissue and not root tissue is that the vascular tissue is closer to the perimeter of, in this case, the stem. It's closer to that. It's not more in the center. That's a dead giveaway. You're looking at stem tissue and it's actually a dicot stem. So if you are an architect and you built high-rise or skyscrapers, you know that you need to put some steel around closer to the perimeter. You don't just have a solid steel core in the center of the building. That would allow for the building to collapse and fall over in high wind. So it makes more sense that the vascular tissue in stems, rather than putting it closer to the internal portions, you put it near the perimeter. Remember, vascular tissue is pretty strong, especially that xylem. That xylem is secondary cell walls, so it's got a lot of lignin, very strong. So if you're stem tissue where you're not surrounded by a bunch of soil that can kind of keep you stable, you're in the air, you're going to want to create some stability. The better way to do that is to put especially your xylem, closer to the perimeter. So that's one thing to look for. If you're getting confused and wondering, is this a root or a stem? Look for that. Now, we said that root tissue, when you look at the dicot root, we said it was this plus configuration of xylem. Phloem is clustered in the crevices of that. But this is, the top picture is a dicot stem. So somehow we have to go from this plus configuration where it's in the center of the root, and then you have to get it so that it's showing this ring configuration in the stem. And I'll show you a picture of that in a minute, but let's think about what we're seeing here. If you work your way out from the outside in, the very outermost region will be the epidermis, especially with a young seedling. You still have that epidermis layer of cells. And then you're going to have some parenchyma cells underneath that. And then you're going to see the vascular bundles in a ring all the way around like that. And then I'm going to point out in between the xylem and the phloem, there's going to be the vascular cambium. And that's meristematic tissue, very precious because it will be routinely dividing to generate more xylem and phloem. So on this image, if you can see the kind of reddish purple where my laser pointer is, I'm hovering over the reddish purple. Do you see that? Okay, the reddish purple, that's the xylem. And then do you see there's this kind of delicate bluish color near my pointer? That's the phloem. And then in between those two, in between the red and the blue, and it forms a ring all the way around, that's the vascular cambium. So it doesn't pop out super clearly because they're living cells, they don't have that lignin, so it's that band of lighter cells, lighter colored cells that you see in between the xylem and the phloem. That's the vascular cambium. So what it's doing, it's going to be routinely dividing, and as it's dividing, as the vascular cambium is dividing on two new cells, one goes to the inside and becomes more xylem. One goes to the outside. And so it just keeps doing that over the life of the growth season, essentially. And you'll get a buildup of xylem and phloem. Right? So the vascular cambium is going to be, over the years, it will lead to increasing diameter of the stem in certain species. In the center then, oh, and so they, yeah, so we call it secondary growth because it, you know, after this seedling has emerged, established, and then the secondary growth is, now we're going to start increasing, in this case, in the diameter of the stem tissue. So it's not primary growth. Primary growth is like when the seed first germinates and you have the shoot elongating, the roots elongating. You don't really have any effort toward increasing the diameter until they get bigger. Sometimes they refer to that central part, that this big swath of cells here, this big section. They often refer to that as the pith, but really it's just parenchyma cells. It's just a bunch of parenchyma cells. All right, so it's nothing special. They just had to give it another name. The image on the lower right is an example of a monocot stem. So that's weird as well. So let's look through this a little bit. Again, we'll have the epidermis right on the outside of the stem. We have a layer of epidermal cells. And then you can see all of the vascular bundles are scattered through the entire stem. They're just kind of scattered throughout. So those are clusters of xylem and phloem, but they're all scattered. And if you look at it closely, there's no way that this has any vascular cambium, like what we see in the image above. There's no vascular cambium. So that means that monocots never truly have secondary growth in their stem tissue to get bigger and bigger in diameter. They don't have it. So that means monocot species tend to be much more smaller in diameter, even at their maturity. And they don't have that true secondary growth of xylem and phloem in the stem tissue. The cortex cells are surrounding the vascular cambium bundle, the vascular bundles. Again, they're just parenchyma cells that are filling in the gaps there. All right, so it's just parenchyma cells. All right, so you got to start training your eye and figuring out which represents the monocot stem versus the dicot stem. And you'll be looking at that again today in lab. And you'll also need to be figuring out, am I looking at root or stem tissue? And you're, especially in cross section like this, you'll need to think about that as well as we work through this material. So this top image is a dicot stem. And we said that the the dicot root has that plus configuration. So that's what you're seeing here. You saw this with the root PowerPoint. So that's an example of a dicot root, where you see that plus configuration of the xylem in the dead center of the root. The phloem are those more delicate cells sitting in the corners of the plus. And it's in the center, though. The entire vascular tissue is in the center. So somehow we have to go from that pattern to this in the stem, where you see a ring of vascular tissue. And I remember first learning, I'm like, how does that happen? That just seems bizarre. But this picture does a good job trying to explain that transition. So down here, if you look closely, there is a plus-shaped drawing here. So this is the dicot root tissue down here, and then you get a portion of the stem called the transition zone, where you will see the vascular tissue starting to morph into a ring of vascular tissue in the stem. So it's gradual, but eventually you'll get, and it's not a long portion of the stem, but you do see this transition, and then you get the full-on ring of vascular tissue in the stem. So I encourage you to look at that numerous times as you're studying to convince yourself that yes, you can go from this plus-shaped vascular tissue in the center of the root to a ring of vascular tissue more toward the perimeter of the stem. So very unusual. This is a close-up of a dicot stem. And again, I'm trying to point out the vascular tissue here. So most of these bigger cells that you're seeing are the vessels, which is part of the xylem. And then you have some tracheids. And then you can see some delicate kind of bluish green cells outside of that. And so it's in between those two that you have the vascular cambium. So it's hard to point out in real tissue. It can be hard to see, but it's in between the xylem and the phloem. And so again, that's going to be pretty regularly dividing and producing more xylem, more phloem, more xylem, more phloem, all through the growing season. But with the monocot stem, there's no vascular cambium anywhere. There's no ring of vascular cambium. There's no vascular cambium hidden away in those vascular bundles somewhere. It's just not there. So that means monocot stems can't get big in diameter. If you look at each one of those vascular bundles, so here's an example of blown up, and they often refer to it as the monkey face, where you have the eyes, nose, and mouth as the vessels, which is part of the xylem. And then the cheeks and chin area are the tracheids, which again, that's part of xylem. And then the forehead is the phloem. So it's some of the monocot stems can actually look very much like that. The next image will show that. So this is a real, I mean, it looks like a face. And so here they're showing, where is it? No, they don't really have it on here. So here, the hat of red, that those are fiber cells that are protecting the very precious phloem right underneath. All right, so sometimes you'll see a hat like that of fiber tissue. Other cross-section of stem tissue isn't quite so pretty to look at. It's not quite that easy to tell, but again, you can kind of see the larger cells here are the vessels, the tracheids. You can kind of see the reddish tracheids, and then the phloem are the more delicate blue-green cells in this image. And then this one's also a monocot. Again, it's not quite as cute to look at. The face is hard to see, really hard. But again, you go by size. So the bigger, huge cells, those are the vessels. And then the smaller reddish cells are the tracheids. So that's all xylem. And then the more delicate blue-green, that's phloem. Here's another cross-section. And again, if you look carefully, you can see the more reddish xylem versus the more delicate phloem. And so monocot stems, because of that scattering of the vascular bundles, there's no vascular cambium, and so they won't get a buildup in diameter of their stem tissue. On the other hand, with dicot stems, you can get a buildup in diameter. And so this is an example of one that's gone through several years of growth. So these are grow rings or annual rings. So if you count how many are there, there's that innermost one that's surrounding the pith. That's just parenchyma cells in the dead center. Then you have this annual ring. So there's 1, 2, 3, 4, right? 4 annual rings. That's all secondary xylem. Okay, so this is 4 years old. Whatever plant, whoever drew this was thinking of a 4-year-old dicot. 4 years of growth. And then they've color coded the red line, the circle there, that red is the vascular cambium. And then the green to the outside of that is phloem. So, you know, I just told you a few minutes ago that the vascular cambium, when it divides, it produces the xylem, the inside, phloem, the outside. So it's like, well, why don't we see an equal amount of phloem? You should see 4 years of phloem growth, just like 4 years of xylem growth. But what tends to happen is the older phloem cells, they're living and more delicate. And usually by the time you have bark on the tree too. And so what tends to happen is those phloem cells will die. They can't take the pressure, literally. And so you only have a narrow band of active phloem for that season. And the older phloem just kind of disintegrates. So that's why you see this unequal, there's a lot of xylem, but not as much phloem. Okay, so don't get too confused by that. But that all that xylem created that 4 years of growth that we see, those 4 annual rings, that's secondary xylem. It's from that secondary growth from the vascular cambium. And that's technically what we call wood. If you look up the definition, it's secondary xylem. So when you cut down a tree and chop up the stem to throw into your wood stove and heat your house, you know, you say you're going to go chop wood, it's secondary xylem that's burning. When you throw that into the stove, it's mostly secondary xylem tissue that is part of the stem, okay? So here is a more realistic example, an actual sample and cross-section. So you have that center where you have the parenchyma cells in the center, and then you have 1, 2, 3 grow rings. So 3 years of growth. And then this is very nicely demarcated where you can see the obvious xylem, and then the vascular cambium is just that narrow band of dark blue. And then out into this region, we're starting to see the more delicate looking cells are phloem. We have some little bit of phloem fibers in there as well, some other parenchyma cells, but don't get too caught up in that. Just realize where the vascular cambium is, you're going to have xylem to the inside and phloem being produced to the outside. And then we're going to get into bark a little bit later because this is also showing bark tissue, but I'm not going to get into that right this second. So another thing to look at too is when you look at all the xylem, you can see that they're different diameters, right? So if you look at this outermost one, you can see that there's some really fat diameter. So those are the vessels. So we would call that spring wood because in the spring, when there's lots of rain, plants preferentially say, I'm going to make vessels, not tracheids. I'm going to do vessels. And so you see this larger amount of vessels in this area. That was during the spring of that year. And then as you start to look further out into that grow ring, you start seeing smaller cells. Those are the tracheids. So that's very typical with angiosperm dicots. They will have the big vessels in the spring because there's lots of rain. They can take up that water and just get it coursing through their body. But then as the summer starts to progress and it gets into dry conditions, drought conditions, more sporadic rain, then they will do the safer thing and start making tracheids because there's less risk of any kind of, if you have big vessels and the plant is moving water through their vessels, sometimes what can happen is an air bubble will pop out of solution and create like an embolism. for the plant, which is not good. It's not good in humans to have an embolism in your vein, not good for a plant either. But if you have narrower tracheids, your risk of an embolism is much lower. And so in the drier season, they'll switch over to tracheids. So that's why you tend to see that pattern of big vessels, and then it tapers down into small tracheids in these grow rings, okay? So we look for that too under the microscope when you're in there today. Here, this is nicely labeled of the different regions. And so the other thing that we'll think about when we get into stems in more detail is that when we think about bark tissue, it's going to be pretty much everything to the outside of the vascular cambium. That's what we call bark tissue. So you see that they have that whole kind of greenish section. They're saying that's the bark. All right. And so technically phloem is part of the bark tissue. It's in that region. But we'll get into that. This is a gymnosperm. So another thing too, if you look here in the xylem, the reddish part, you'll see that they all pretty much look the same size in diameter. So let me go into that. Where is it? Here. This is a gymnosperm, and you can see how the xylem is pretty uniform. All the same diameter. So that's a dead giveaway. You're looking at a gymnosperm, because gymnosperms don't have vessels. They don't have them. Well, there's like a couple of plants in the gymnosperm that have them. They're really weird gymnosperms. The vast majority of gymnosperms do not have vessels. So these are all tracheids. They're all the same, pretty much the same diameter. So that's another thing to train your eye to as you're looking at the stem and cross section. You're going to look at monocots and dicots today, and some gymnosperms as well. And then the other thing you're going to look at are lenticels. Lenticels, we learned a little bit about that already, but lenticels are areas of loosely packed stem tissue. And it's just loosely packed so that you can get some gas exchange going on in those regions. Because stem tissue, all the living parts of the stem have to get access to oxygen to do their thing in terms of ATP production. So in cross-section, you would see something like that. So we've seen this already, where you have that loose configuration of cells. Those are lenticels. It allows for the stem tissue to breathe, so to speak. So different designs depending on the species, but it's usually fairly noticeable under the microscope. Sometimes it can be hard to spot if you just walk up to a tree and look. It can be hard to find. Here on birch, it's really simple to find, but other species, it's challenging. Then you'll also look at some modified stem tissue today in lab. And we have some fresh samples of corms and bulbs and rhizomes. And so thinking about those ideas, again, it's learning the term and its definition. And so you'll be working through this in lab as well, okay? So as you are examining all these in the lab, start linking that together with the term and the definition. So trees or plants have a good way of modifying roots and modifying stems in order to serve their needs. A lot of it has to do with storage of nutrients, okay? All right, so lots of things going on today in lab with regard to stem tissue. All right, so I want to switch back now into the slide set on photosynthesis and wrap up the last topic on there. So we've been covering the C3 cycle, the Calvin cycle. We looked at the idea of how sometimes rubisco is really okay with oxygen coming into its active site, and how that can cause problems because it can kick off the PCO cycle. So we looked a little bit at the PCO cycle and realized that if a plant does the PCO cycle, not the greatest thing to keep doing because you can't feed both products right into the Calvin cycle, the C3 cycle. They won't, the PGA will fit, but not the 2-carbon phosphoglycolate. And so the plant has to send the phosphoglycolate into this complicated PCO cycle. It's complicated, takes time, takes energy, ATP gets used up, CO2 gets given off. That's not good. So all in all, not the best situation. And this tends to happen under more drought, hotter temps, drought conditions. That's when the plant will start closing down its stomata. And if a plant does that, they're not taking in CO2. That means inside the leaf, the CO2 levels drop lower and lower and lower and lower. And so pretty soon, Rubisco's like, where did all the CO2 go? It's gone. I only have oxygen around me. So then oxygen will win and come in and react with RUBP. So under those conditions, C3 plants will kick into the PCO cycle. And again, it slows down their growth, slows down their ability to reach maturity and form flowers and fruits. So then we learned about how some plants are able to do the C4 cycle. Those plants that are in hot conditions pretty much year round, and they get rain, but they do go through periods of drought. And so under those conditions, they evolve the C4 pathway, the C4 cycle. So that's what we're looking at here. We worked through that. And then we looked at the idea how with the C4 cycle, there's different things happening in different places inside the leaf. And so you have some things happening in the mesophyll cell and some things happening in the bundle sheath cell. So it's spatially separated, they say. All right, and so the mesophyll cell is where it all kicks off, where the CO2 that's being brought into the plant through the stomata will react with PEP, this molecule PEP, and it's in the active site of the PEP carboxylase enzyme. So that's not Rubisco. So that CO2 coming in doesn't work with Rubisco right away, but that's okay because PEP carboxylate is an awesome enzyme. It's able to only work with carbon dioxide and PEP, no oxygen. So then we get a reaction that produces oxaloacetic acid, which then converts to malate or malic acid. And malate has a very important job because it has to deliver the CO2 that's part of its body, has to deliver that over into the bundle sheath where Rubisco is waiting. And so Rubisco's in the bundle sheath, that CO2 will get fed to Rubisco, which is great because that will allow the Calvin cycle. Once that happens, you're left over with pyruvate. That's okay. That can go back into the mesophyll cell and reform the PEP molecule. And so it is a cycle. You got to get more PEP ready to go. So as CO2 comes in, it can react with it. But this is super efficient. And yes, there's an expenditure of ATP. You do see that right on here. There is an extra expenditure. So it costs more to run the C4 cycle. But if you live in tropical areas, it's worth it. You're very efficient at photosynthesis. Now, if you have them up here in the temperate region, C4 plants, they actually are at slightly a disadvantage most of the time here because they're expending ATP when they don't really need to because they have wetter conditions, cooler conditions. But if you evolve to live on the equator area along that region, like corn, maize, then it makes sense to do this pathway because you consistently see fairly hot conditions year round and you get drought periods. So this makes them very efficient. Questions on the C4 cycle? Yeah, I'm about to talk about next though. And the good news is, it's not a whole different cycle yet. So this is going to look familiar in a second. I'm going to talk about succulents. And some of this is going to look really familiar. But succulents, they do a really interesting innovation that they evolve. So what they do, So we said how C4 plants do this spatial separation. Something happens in the mesophyll cell, other things happen in the bundle sheath cell. Well, with CAM plants, which succulents fit into that, we call them CAM plants. Oh, do I have that spelled out somewhere? Let me look. Yeah, I don't see it spelled out. It's in the book. It's the C stands for Crassulaceae. Crassulaceae. Big long word. That's coming from the family of this group of plants. Crassulaceae. That's the C. A stands for acid, and M for metabolism. Crassulaceae acid metabolism. Or now you know why we just say CAM, because it's a pain to say that over and over. So CAM plants, you can see some of the same players in this pathway as what we were just talking about in the C4. But they don't do a spatial segregation of where things are happening. Everything happens in the same cells. But you see a temporal, they call it a temporal separation, time separation. So they flip when they are doing certain things. Some things happen at night. Some things happen during the day. So if you are a gorgeous giant saguaro cactus living in New Mexico or Arizona, and if you've ever been there or know something about those states, it is hot year-round, pretty much. Long periods of time. If you're a gorgeous giant saguaro cactus, would you keep your stomata open all day long? No, you must be gorgeous. All right? Because drought is a thing. It doesn't. I'll read that. So it's going to be very challenging to access water consistently. And yes, they have monsoon season for sure, but that's temporary. They, you know, it comes in bursts for several days and then it stops. Long periods of drought. So if you want to stay alive under those conditions, don't open your stomata during the day where water is leaving your body all day long in the sun. Don't do it. So what they do at night when it's cooler, a little bit more humid, they'll open the stomata. That's when they bring in the carbon dioxide. So they'll open their stomata at night, bring in the CO2. So that's what you're seeing on the right side of the slide. And then what they're doing is they still react with PEP. So the CO2 and the PEP, yeah, just like with C4, same enzyme, carboxylate. So it's all the same. It's just happening at night. So now they're making malic acid. They make OAA and then malic acid, just like what we saw before. But now they got to store up that malic acid all night long. So what they do is they stuff it into their vacuole. And you can actually insert a pH electrode into the vacuole all night long, and you'll see the pH drop more acidic, more acidic, more acidic all night long, because malate is an acid. It's malic acid. All right, so that's why they call it CAM. The A stands for acid. So all night long, this vacuole is getting more acidic because malic acid is getting stuffed in there all night. Because remember, the Calvin cycle can't run at night. So the CAM plants have to like, okay, I'm gonna take all the CO2 in all night long, but then I gotta store it up because my Calvin cycle is not running. So then when the sun rises, they're gonna close those stomata really quickly. And now the job is to get that malic acid mobilized out of the vacuole and get it over to the chloroplast where it can get the carbon dioxide cleaved off of it. So the malic acid, again, is still delivering. It serves the role of being a delivery person, of delivering that CO2 from the vacuole into the Calvin cycle where Rubisco is waiting. All right, so during the day, you got to get that CO2 over to the chloroplast where Rubisco is so that you can get the Calvin cycle up and running. So it makes a lot of sense, again, if you think about these plants having to survive a desert condition. It makes a whole lot more sense to open your stomata at night so that you're not losing all this beautiful water out of your body. And so again, the payoff is still in the idea that Rubisco is getting surrounded by a bunch of CO2 all day long, with that malate coming out of the vacuole all day long and delivering the CO2 right to where Rubisco is inside the chloroplast. So this is all happening in the same cell. There's no spatial like mesophyll bundle sheet stuff going on here. It's a day-night rhythm that we see certain things happening during the night, certain things happening during the day. So again, this is just verbal or written down what we just went through. So of the three groups of plants, so we have C3 plants that just do the C3 cycle. Then we have C4 plants, which tack on the C4 cycle along with the also on the C3 cycle to do both. And then you have the CAM plants that do CAM and C3 cycle. So if you look at those groups of plants, the CAM plants win in the sense of they are the best at conserving. So they're very, very good at holding on to water and still running their CAM pathway that links into the C3 cycle. So they're really good. And then C4 plants are the next best at conserving water. And then the C3, they rain blast. They're a little bit more luxuriant with their water loss. CAM plants that you may be familiar with, pineapple. So growers that have pineapple plantations, that's they have to think about these ideas. So succulents, many succulents, but not every single succulent necessarily does CAM. So unfortunately, they named them CAM plants before they realized that. So just because it says Crassulaceae, not every single species in that family, Crassulaceae, necessarily does the CAM cycle. So there's no 100% they all do it. This lower right or lower left image is a weirdo. This is an actual gymnosperm. So this is a weird gymnosperm that can actually do CAM photosynthesis. Yes, very much. And we're talking extreme drought, extreme heat during the day. So there are some plants, there are a few CAM plants have been discovered that can turn off the CAM pathway. So there's not many out there, and I don't have them memorized which ones those are. So when I have some succulents in my bedroom, for example, and I want them to take CO2 in all night long because that's cleaning up my air quality in the bedroom. And so I want them to do the CAM pathway. So I say I'm mean to my CAM plants. I don't water them luxuriantly, because if you give a CAM plant that can turn off the CAM pathway, that's how it would happen. You would keep watering your plant consistently. Then it's going to say, wow, I'm getting all this lovely water. I'm not going through 10 months of drought. I think I'll just turn off the CAM pathway because it's extra work. And so I don't do that to my succulents. I'm stingy with when I water. And in general, it's stingy with water. It depends on the pot size, but generally if I have like a 6-inch pot with a full grown, it's a mature succulent, every 3 to 4 weeks I'll water. And then sometimes I go a little too long, and then you can actually start seeing the succulent kind of getting wrinkled because there's so much water loss that it's like shrinking. All the cells are shrinking so much that it gets wrinkly. You don't want that to happen. Yes, I do. I do that. So like I have a lot of plants in my office right now, and I'm generally pretty stingy with watering. I don't fertilize in the winter either. I want them to kind of maintain that rhythm of, that this is their sleepy time. And then I'll wake them up in the spring and be like, okay, here's some fertilizer, repot, give it fresh soil. Yeah, but in the winter, I like to simulate, kind of go work with the process of evolution where they're a little bit more dormant in the winter. So So yeah, lots and lots of people that have come to me with plant problems. It's often succulent, and they're too nice. They actually, like, I had a person watering their succulent every day because they wanted to be nice to their plant. I applauded that they cared about their plant. That's great. But I had to convince her, be a little mean, be a little stingy with the water. They really don't need that much. So again, if you have an understanding of how they do photosynthesis, where they evolved, I think it gets you a little bit better understanding with your houseplants too. You can figure out which ones really do need consistent water and which ones you can let the soil really get dried out before you water. Okay, so it matters what kind of photosynthesis they do for sure. And if any of you have gone into desert situations, you could hear the giant saguaros, which I believe are, well, they're the state flower, the flower from the saguaro cactus. They're the state flower. And I believe they're protected. Like if you have these on your property, if you live in Arizona, you can't cut them down. It's against the law. You might be able to get them moved, but you have to have special people come in and move it. So they're protected. Gorgeous though. I mean, look at that. That's just fantastic. So if you've had the good fortune to wander around in a place like this, that's great. And next time if you go into an area like this again, I hope you can bring this knowledge with you and think about all these plants and what they're doing at night. They're very busy at night. So, okay. In EduCat, I've posted a sign. It looks like a quiz, but it's not. There's no time limit on it. Open book, open note. So it's gonna start, it'll open up today. You get one. And you'll have a week. So it's in the first top portion here where you see exam one. It'll be in that section. All right. And so this is homework to get you to work through some of the concepts that we've been talking about with photosynthesis. And you do have to get these concepts straight in your head on what's happening. Recycle, okay, then layer on what four cycle accomplished. So knowing the cycle for sure. There's going to be key things you need to know, and then understanding what types, where would a plant live doing this kind of stuff. So putting it into context of where do they evolve. So let's look at this a little bit more. Let me see if I can. Hang on just a sec here. Well, hang on. I'm going to get into this a little differently. Hey, boy, there we go. Okay, so I'm gonna draw out something for you here that I want you to follow along. So pull up on your laptop, if you have your laptop, the C3 cycle, Calvin cycle or the C3 cycle. So when you look at that, it's a busy slide and it has, you can count the carbons and all of that. But what are we really aiming for in terms of your knowledge for the Calvin cycle? So if you think about the process, we have RUBP is going to react with CO2. All right, so CO2 and RUBP are reacting. Sorry for my sloppy writing, but if you have the slide next to you, see. So what's the enzyme for that? Rubisco, CO on the end, all right? So you have to keep straight that Rubisco is the enzyme, RUBP is the 5-carbon molecule that CO2 reacts with. So Rubisco is sitting here in the chloroplast, in the stroma, and RUBP comes into its active site, CO2 comes into its active site, and they react. You get a temporary 6-carbon molecule that quickly breaks down, right? So don't worry about the 6-carbon. What does it break down into? The two identical molecules of what? You can say the abbreviation. Yeah, PGA. So on the homework assignment, if I ask something like that, you can actually just put PGA. You don't have to spell it out, okay? Probably on the exam, I'll spell it out plus put the abbreviation, just to be clear. So that's a really important step because this is the first step of the Kelvin cycle. It's what kicks off the whole thing. So yes, I want you to know it to this level that we've drawn. So now those PGA molecules, each one of them will go through some more reactions. Wow, that's weird. So I'm just going to put arrow, arrow. And we know that ATP is needed in these steps, and we know that NADPH is needed in these steps. Where is ATP and NADPH coming from? How do they get made? The light reactions, you need to know that. They're coming from the gorgeous light reactions cooking along. So PGA gets finally converted to another, we have a 3-letter number acronym for it. What is it? G3P. Okay, so let me just emphasize, please get these straight in your head. I've seen students mix up PGA and G3P. They put them in the wrong spot. So somehow figure that out when you're studying to come up with some silly saying or acronym where you know that the first stable product is PGA, and then you go through a series of reactions that need ATP, need the NADPH, and then you end up with G3P. So this is a good point in the cycle because the plant can take advantage as the G3P is more and more of that gets made. They can take some of that and siphon it off into another pathway. And what are they going to do with that G3P in that other pathway? What gets made? Something the plant really wants made. It's very important. Sugar. Sucrose is actually not glucose. That's a common mistake. People think it's glucose. It's not. It's sucrose. So they're going to be making sucrose in that other pathway. I'll just put SUC. So that's great. But now we need to finish the cycle because it's a cycle. So we got to take it back to the start. So then we're going to see, again, a bunch of stuff happens, bunch of reactions. And toward the end of a bunch of those reactions, you have to expend some ATP again in order to get that ribulose phosphate. It needs one more phosphate, so it can be called RUBP. The B is bisphosphate, that means 2. So RUBP has 2 phosphates. So again, you have to expend ATP to get that to happen. So what we drew here, that's what I want you to know for the exam. If this is simpler for you to study off of, great. The other diagram taken from the book is busier and shows you can count carbons and convince yourself of what's going on carbon-wise, which is fine. Okay, so the C3 cycle, you do need to know the reaction steps and when ATP is needed, when NADPH is needed. Questions on the Kelvin cycle? Yes. ATP, yep, so ATP feeds into this. NADPH feeds into this, yes, yep. So take that drawing, hopefully you drew this along with me, and then compare it to the one in the book that's busier, and I think it'll start to click in place. On next Tuesday, we'll do a short review with C4. I'll do this again with C4 and CAM, get you thinking about, what's the key stuff here, all right? And then we will also come back to roots and stems next week and get into leaves as well, all right? So lots of stuff happening next week. All righty.